So in terms of the mechanism of DNA replications, we do know that for Manstein and Stoff experiment, DNA replication is by semi-conservative. So means that this proposed by Watson and Crick. Watson and Crick, these two persons, they were the ones that found out the shape and the structure of DNA. So in this model, the two parental strands must unwind, so serve as the templates. So this result in the formation of two hybrid DNA molecule with one parental strand and one newly synthesized strands. So what are the three main processes here? We have to unwind the parental strands, forming the leading strands and also the lagging strands. Okay, so we need to know what are the enzymes involved in the unwind of the parental double helix. So DNA replication begins at a specific site. So this specific site is known as origins of replication. This origin of replication can be recognized by helicase. So the helicase will bind and then start to unwind. So what helicase do? When the unwind takes place, the Y-shaped region is known as replication fault. Okay. So first enzymes, we need to know helicase. What did helicase do? Helicase destabilize enzyme. Okay, destabilize the DNA molecule by breaking the hydrogen bond. Highlight. Thereby separating the two strands. So it travels along the helix, opening the double helix like a zipper during the movements of the replication forks. So with hydrogen bond between the complementary base pair are broken. Okay. Now another enzyme involved here is called topoisomerase, not in our syllabus, but come out in the paper one before. But based on the answer, you will be able to answer the question. Are you clear? The choices there. So topoisomerase, okay, don't worry about this, because when you try to unwind something, you pull the string. Try to imagine that the two strings are already wind already. You want to unwind it by pulling like this. At a certain moment, you won't be able to unwind them anymore. It creates tension there. Are you clear? You try to imagine that you wind something and then you pull it. You pull it this way, at a certain moment, you will no longer can actually unwind anymore. So this creates tensions. Topoisomerase help to release all the tensions. Okay, so generally, it enables tension caused by the unwinding to be released. Okay, topoisomerase, not in our syllabus. Okay, then we have this single strand binding protein. Single strand binding protein actually to prevent the double helix from reforming until the strands are replicated, because double helix, it will form back this, okay, form back this hydrogen bond. So the unwind sections of parental DNA now available to serve as a template. However, the enzyme that synthesizes DNA cannot initiate the synthesis of the polynucleotide. So this enzyme right now is DNA polymerase. So DNA polymerase they do not know where to start first. Okay, so therefore we need to add a DNA nucleotides to the end. The already existing chains, they only add the DNA nucleotides that is base pair with a template strand. So means that they only recognize the double-stranded structure rather than single-stranded structure. So the initial nucleotide change that is produced during DNA synthesis actually have a short strand of DNA known as primer. Now, primers, the understanding of primer is not in this topic. The understanding of primer, just now I said the RNA primer is in next topic and also in our A2 syllabus. Are you clear? Okay. So the primer is synthesized by the enzyme called primase. It's a single RNA nucleotides. Okay. So the new DNA strands will start from the 3' ends of the RNA primer. So just now I show you guys the 5' start first, right? So how we join? We join at the 3' end and continue. Okay. So the entire process unwind the parental double helix. What is the first process? The first process, helicase unwind and separate the parental DNA strands by breaking down the hydrogen bond between complement-based pairing. So make the parental strand available as template. This is how we write, okay, for the first process. So what is number 2? What is number 2? Number 2, we have the single strand binding protein stabilize the unwound parental strands, hence prevent the parental strand reforming complementary base pair or reform the hydrogen bond. Okay. So primary synthesized RNA primer using parental DNA as a template. So DNA polymerase now can add free activated DNA nucleotide to the 3' ends of the RNA primer. Okay, so let me show you guys something. So guys, this is the parental strand. Okay, parental strand. So parental strand here 3', here 5'. So when we add the primer will start with 5'. So this part actually is a 3'. Can I see that? So means that DNA polymerase is going to add the nucleotide one by one here. Join, one by one. So it join one by one like this. So can you see that directions is from 5' to 3'? Are you clear? Always remember synthesis must be 5' to 3'. Okay. So synthesizing a new DNA strands, the enzyme involved highlights DNA polymerase. Okay, what they do, they add, highlight free activated DNA nucleotide. In AS syllabus, we say free activated DNA nucleotide. In A2 syllabus, we can use DNTP. Okay, so what's DNTP? Deoxynucleotide triphosphate. They have 3 phosphate groups, so that's why it's activated, right? If not activated one, no, wrong, guys. If you say that 3 DNA nucleotide, wrong. If you say activated DNA nucleotide, also wrong. You have to use free activated DNA nucleotides, must be in full. So this DNA polymerase require a primer, as I say that, and a DNA template strands, along with a complementary free activated DNA nucleotide. So they add one by one, which is complementary, okay? DNA polymerase can add free activated DNA nucleotide only to the free 3' ends. So highlight the direction must be 5' to 3' direction. And this free activated DNA nucleotide or DNTP, they have triphosphate group. Are you clear? When you remove the pyrophosphate, means that 2 phosphate group, energy is released. Okay, so this energy is exothermic reactions. So helps to drive the polymerization reactions by forming phosphodiester bond. Are you clear? Okay. So in this diagram, can you see that, guys? Reactions start from 5', continue all the way in 3'. So the nucleotide is a free activated with 3 phosphate group, remove 2 phosphate group, then form the phosphodiester bond. The enzyme involved here is DNA polymerase. Okay. So due to the anti-parallel arrangements of the double helix and mandatory 5' to 3' reactions, so you're going to form leading and lagging strands. If you say leading strands, DNA polymerase remain in the replication forks, so they will process will elongate continuously. So the whole process only require 1 primer. Are you clear? But lagging, DNA polymerase must work along the opposite template in the direction away from the replication forks, and this was synthesized discontinuously, again direction 5' and 3' in the segment, and this segment is called Okazaki fragments. So means that we need to prime separately. You cannot use 1 primer and continue. We must continue to add primer. Okay. Now E, no need to worry. E just for information only. We need to remove. After DNA polymerase form an Okazaki fragment, another DNA polymerase replace the RNA nucleotides of the adjacent primer with the DNA nucleotides. Okay, but it cannot join together. So we need to have another enzyme known as DNA ligase join the sugar phosphate backbone by forming, right now, guys, by forming, okay, phosphodiester bond. Okay, by forming phosphodiester bond. Clear so far? So this one we have break down, okay, the process, okay, 1 and 2. So after RNA primer is made by using the parental strand as template, DNA polymerase starts to add complementary highlight free activated DNA nucleotide to synthesize a daughter strand. Okay. So the newly synthesized daughter strand elongates towards the replication forks in the direction of 5' and 3', forming a continuous leading strand. So leading strands always have no problem. Are you clear? always has no problem because you only need to prime one time. But how about the lagging strands? So lagging strands, you can see that we must have this, okay, DNA polymerase, short primer one time, Okazaki fragment 1, Okazaki fragment 2. Then after that, you can see that we need to have the ligase to seal the sugar phosphate backbone. Okay. So we say that after RNA primer is made by using the parental strand as template, DNA polymerase now start to add complementary free activated DNA nucleotide. Direction still the same, always 5' or 3', but this time because antiparallel, it will be away from the replication fork, forming this continuous Okazaki fragment. So in this case, I formed the Okazaki fragment 1 first. After reaching the next RNA primer to the right, DNA polymerase detached from the parental strands. Okazaki fragment is now completely synthesized. Can you see that? Here, continue. Oh, stop. Right? So fragment 2 is primed by RNA polymerase. Again, DNA polymerase add free activated DNA nucleotide in the direction 5' to 3' directions, again away from replication fork, forming Okazaki fragment 2. So DNA polymerase again detached from the parental strands when it reached the fragment 1 primer. Another type of DNA polymerase. Okay.
你之前那不是看的挺欢的吗？那个
哪来啊？就剩我一个人在看都不看。
Okay, so we have another type of this DNA polymerase. Remove the DNA primer by adding the nucleotide at the 3' end of fragment 2. So this one, no need to know. Okay, later I will teach you guys how to write. Okay, then we need to have DNA ligase to seal the sugar phosphate backbone by forming this phosphodiester bond. The lagging strand is formed discontinuously from 5' and 3' away from the replication forks. Okay, so now no worry, I'm going to summarize. Okay, so how you write? First, unwind. First process. Unwinds of the parental double helix. Okay, so how we unwind? So first, process number 1. So we need the helicase. So we say helicase unwind the parental double helix by breaking the hydrogen bond between complementary base pair. So once you unwind ready, so number 2, now parental strands can act as templates. It's exposed and form templates. Now, to prevent the parental strands to actually join back, okay, to form back the hydrogen bonds, then we need to have the single strands binding protein. Okay, prevents the formations of double helix. Okay, by the time prevents the formations of double helix. Okay, so this is the first part, unwinds of the parental double helix. Clear or not so far? Okay, process number 2. Formations of the, leading strands. Now, as I said that just now, primal, the concept of primal is not required in the AS syllabus. So whether you talk about it, you get the, I mean, I don't say bonus mark because to describe the process may be 5 marks. So you need only 5 marking points to get 5 marks. But sometimes if you miss 1 marking point, but you mention about the RNA primal, sometimes you get the marks. So it means that if you forget some point, you add it in the other part. You still can get that mark, but you do not need to put that in if you have to complete what I have written here, the minimum. Are you clear? Okay. So I will use green color. I will use green color. Green color means that it's not necessary for you to put in. Are you clear? Okay. So first process, primase, synthesize RNA primer, or just primer. This is the minimum to write. Okay. So what is the next process? Number 2. Okay, I will say enzyme very important. DNA polymerase. Add. Okay, complementary. Free activated DNA nucleotide. Okay. Forming. Okay, sorry. Nucleotides. Sorry, add the free DNA nucleotides from 5' and to 3' and forming the continuous. Oops, sorry. This one a bit long. 2 words. Replication forks. Okay, so DNA polymerase adds the complementary free activated DNA nucleotides from 5' end to 3' end towards the replication forks. So 3 leading strands is synthesized, okay, continuously. Okay, forming a continuous leading strand. So this is formation of the leading strands. Very simple and quick one. Are you clear when you write? Now, number 3, formations of the lagging strands. A bit tedious, more thing to write. Formations of lagging strands. So first, you still see the same. Okay, primase synthesize. Okay, primer. Whether add in or not, up to you. Number 2. So same thing. We say DNA polymerase add free complementary free activated DNA nucleotides. Direction still the same from 5' end to 3' end. Okay, away from a replication forks. Okay, away from replication forks, forming Okazaki fragment. Okay, so DNA polymerase adds complementary free activated DNA nucleotides from 5' end to 3' ends away from the replication forks, forming Okazaki fragments. Number 3, DNA ligase seals the sugar phosphate backbone okay, DNA ligase seal the sugar phosphate backbone, okay, by forming okay phosphodiester bond. Okay, so lagging strands is synthesized discontinuously. It's minimum. Enough already, guys. 5 marks for DNA replication. If you include everything, you should get more than 5 marks. The green color item not necessary. Are you clear? Okay. So what you need to know and what you need to write in the exam, you can see that's a huge difference. Yeah.